Next, let's talk about new technologies for detection of the HER2 gene alteration. I understand that at ASCO there was some update on next generation sequencing methodologies to detect HER2. Can you comment on those abstracts, please?
Well, there were a couple of abstracts looking at next generation sequencing, trying to make the point that maybe we could more accurately, assign HER2 status based on next generation sequencing. They found interesting cut points and all, but I think what we haven't seen is, would next gen sequencing do any better at picking out a population who benefits from HER2 targeted therapy? So maybe we can define levels of amplification and overexpression, but the real thing that matters is, are they going to respond to HER2 drugs? I think one of the most important things that next gen sequencing can do is to find an HER2 activating mutation. And as we know, that happens probably more commonly in the classic HER2 negative population, and yet we've seen responses. We've had trials, certainly with neratinib, in a classic HER2 negative population, but with an HER2 activating mutation, that a tyrosine kinase inhibitor to HER2 can have efficacy. So I think from my perspective right now, where I would use next gen sequencing is, I'd be looking for an HER2 activating mutation. I did have a recent case where it was present in a HER2 positive early stage patient. She was enrolled in a clinical trial of trastuzumab and pertuzumab, a TBCRC trial, and had no response at all to trastuz pertuz. Didn't make any sense to me. Her tumor highly overexpressed HER2. Then I added chemotherapy and had some response. And at that point, at the time she went to surgery, we actually ran next-gen sequencing, found an HER2 mutation, which I think explained why she hadn't had a hint of response to the HER2 antibodies. So this is something to look out for. I think a problem with next-gen sequencing is, I think we're learning that HER2 heterogeneity is important. And in some recent trials that we've seen, the populations where there's a clear population of tumor cells that are HER2 negative, that's 30% or greater, they don't do so well with HER2 approaches. Also, this HER2 2+ and FISH positive population might not be doing so well as the HER2 3+ when we look at the results of some recent trials. So I think we have a lot of work to do, but as we define the way that we are testing for HER2, I think we have to remember that the bottom line is that we are doing it so we can assign treatments. There's been some exciting early data that maybe trastuzumab deruxtecan might work better in a HER2 lower population. But we had, the NSABP B47 trial that enrolled the HER2 1+ and 2+ FISH negative population that showed that trastuzumab didn't work there. Maybe we need to repeat that trial with trastuzumab deruxtecan as well. And actually, we have a very interesting trial going on with our colleagues at the Uganda Cancer Institute, looking at using a platform that's commonly available in sub-Saharan Africa that they use in terms of TB and HIV analysis. So the platform's there, and they've got a cartridge that can do RT-PCR on tumors for ER, PR, HER2. So you don't need a complicated lab set up to do immunohistochemistry or FISH. And we're comparing kind of this African version that is very practical, very doable, with the gold standard that we do in Seattle right now, and are finding a very tight correlation. So for rest of world that doesn't have access to these labs with not just next-gen sequencing, but a lot of places that can't do immunohistochemistry or fluorescence in situ hybridization, we might have some interesting new assays here as well.
That's really interesting that you bring up the issue of this real-time PCR cartridge. That's actually from some colleagues that are nearby us here in Northern California in Sunnyvale from Cepheid
Right.
Corporation. And in their paper, Natalie Wu and Mike Press was a co-author on that paper. It's really interesting if you look at their, HER2 low subgroups, that is the patients that are 0 or 1 plus by IHC. If you look at their transcript level, it's not dissimilar to HER2 2 plus FISH negative. So I wonder in the development of trastuzumab deruxtecan, which currently the HER2 low definition is IHC 1 or 2 plus FISH negative, I wonder whether we should also include HER2 zero IHC based on these transcript levels that
Yeah.
look about the same as 1 or 2 plus IHC. I think that could be a missed opportunity, and we should look into that in future clinical trials indeed.
Let's partner in a trial.